So back to Zina then. So, yeah.
Well, you see, so I published a paper, not in
87?
87?
I think with Zina, yeah.
Yeah, no, but I published a paper in 1982.
Okay.
How does extracellular matrix signal is 1982.
Okay, that's
okay.
Yeah, before.
So I published that extracellular matrix probably signals, and I had written it in 1980 before integrins were known, and it came out in January 1982 in Journal of Theoretical Biology. That is one of my most important papers. So I postulated that extracellular matrix had qualities that people had not recognized. People were beginning to think that maybe there should be a receptor.
Receptor, absolutely.
And I said it had to have a receptor, and it had to signal through the cytoskeleton, and the cytoskeleton had to talk to the nucleus, and the nucleus had to talk to chromatin and had to reorganize it. And when it reorganized, then the nucleus would start signaling to outside, and that changed outside, and I called it the model of dynamic reciprocity.
Yeah.
And people had used this reciprocal thing between outside and inside, but nobody had even come close to say that this has to be a continuum, and it has to go all the way, because otherwise it doesn't make any sense why we do what we do. Why 10 to 70 trillion cells in your body with the same genome make your nose or your mouth or your breast or prostate or whatever?
What they are. Yeah.
How do they do it? So I said that extracellular matrix could be a very important component of that signaling thing. And it has to start very early, and it has to tell the tissues what to do. So we then published, few of these other papers in whatever. And I was in a meeting, Rabia Talhouk asked to come to my lab. And Zina and I were good friends.
Oh,
Zina
so you knew each other, okay.
Well, Zina
she
was
in
at
California.
UCSF.
Okay.
She was at UCSF. I was in Berkeley. And a student of Zina, who sadly died of cancer later, wanted to always come to my lab and think about doing these things. And we, in fact, have a few papers together. And so Zina and I would go to meetings together, like later on to Integrin meeting. Richard Hines was once in a meeting where I gave my first thing with the postulate.
Richard Hines from MIT, who is at MIT now.
Yeah.
Okay.
Yeah. Few years after that, integrins were discovered, and everybody, So he once heard in one of these Gordon conferences about my work about the fact that function changes when you put, and he said, Mina, this is important, I got it. Then he did his own Gordon conference and invited me. And Zina and I were in that, I think. And in any case, she said to me, I love the way you do science, you're so original. And we were friends, whatever. And she's such a good scientist herself. And but she was working with fibroblasts and rabbit fibroblasts and metalloproteases, okay? So at one point she said, I really like to collaborate with you. And so when Rabia wrote me to come to my lab, and I decided I always go back to the animal and go back to the normal to actually find out what's going on. So I said to Rabia, we can't do any of these things unless we take the work into the mouse. And it's one thing to take the mouse and do all these studies in whatever, but we have to find out what happens in virgin pregnancy, lactation, involution. So he started doing these experiments, and he discovered that a couple of metalloproteases. So then we said, Zina, I teach you mammary gland biology, and we learned some MMPs from you, and that was the basis of 20 years of very, very good collaboration. We now have decided to separate, but it was time because,
So, back to that review that you came with the word dynamic reciprocity. How do extracellular matrix and hormones direct
No.
gene expression.
The hormones.
That was the title.
Okay. The hormones, I wrote another review in 1987.
Okay. So that was the idea.
The first dynamic reciprocity
Came
review
earlier.
was in 1982.
Oh, that's the Journal of Theoretical Biology. Okay, so it
All
was
right.
not in the title, but it was in the text, probably.
What?
The
dynamic
probably.
No, it says, how does extracellular matrix signal?
Okay.
And I wrote a review the year before that, and I said, what is the definition of a normal cell in tissue culture?
Mm-hmm.
And I said, you have to go back to in vivo, and if you read everything, you see that everything gets lost in culture. Mm-hmm. Then I did that Journal of Theoretical Biology with Glenn Hall, the guy who taught me about basement membrane, and Gordon Perry, who was a British guy who was helping me, who was a postdoc, and then he hasn't done, he went to a company after, but really, basically, I just sat there and it hit me that it had to be like that.
Absolutely.
And so every step of that model, one way or the other, either in my lab or other people's lab, actually has proven to be correct, which is really lovely. Mm-hmm. And one of the first things I tell to my students on postdoc, try to disprove this model, try to disprove me, and then design your experiment to disprove everything. And if you prove it, then do it again, and then do it again, and then you could,
be confident that.
Yeah. So then we started collaborating with Zina, and our later paper had something to do with Sharon, the student that Zina had. But the first real paper we had with Rabia was, in the pattern of MMPs and everything else in whatever. We had a paper, I think, so Zina and I decided we would switch off regardless of who had done what. And so our first paper was in development, then in Journal of Cell Biology, et cetera, et cetera, et cetera. And then it just mushroom from there, and the mouse of the MMP3 mouse was actually made in my lab. Mm-hmm. And I was on sabbatical, and I said to Zina, write it, and you can be the senior author, and that's how it was, and it just was very good.
Fantastic.
Collaboration, and we learned a lot from each other, and the group benefited.
Yeah. So it's in the early 80s that the field of cancer genetics progressively outpaced the view of cancer as a systemic disease involving non-self-autonomous components. What have been the consequences of this tumor cell-centric view?
Well, you know what has been the consequence. They didn't have very many rebels, and I think the problem was that genetics and the molecular biology and the concept that genes and the sequence of DNA and oncogenes and suppressor genes, it was so sexy and so tempting and so wonderful, it was a very important, but people just immediately discarded aerobic glycolysis, discarded all this work that has been done over hundreds of years, actually. This work in wounding, there's, the whole model of two-stage carcinogenesis, initiation and promotion of tumor promoters. The TPA field was huge in the 70s, and that all got thrown out. I mean, in terms of the top journals, in terms of, and I think one of the other problems. Now, this is a really different point of view that many people have, is that a lot of physicists came into biology, and they are incredible reductionists. And when they came to biology, especially in the 60s and 70s and 80s, they could do a lot of things with simple models and simple gels, and things could get explained. And I mean, if you read some stuff of the 1970s and 80s of what was going on at Harvard University by a guy called Wilson, who is a great entomologist, probably the world's greatest entomologist. He has a huge, beautiful textbook, and he was a professor, thoughtful. But he writes in his memoirs that that the molecular biologists at Harvard were pushing everybody in physiology and system, the evolution, blah, blah, out of the door, and they were going to, they were coming and measuring their labs so that they can build their own lab, and they would, and it became a very elite bunch of people who were interacting with physicists very fruitfully, but also under the light. They were looking at the next step and never at the larger questions. That in my mind, if you are really want to change the field, if you want to make a lot of new discoveries, or you want to change, you have to shift paradigms. And they were not at all susceptible to wanting to be wrong. And I told you, they still don't. Okay, there is still a group of people who honestly are, even though they mount the word microenvironment, they say, oh yes, complexity is important, whatever. In reality, they all work on 2 dimension, or if they, whatever, they do these huge data sets, which are fantastic, but they're in lousy systems. Yes. And so it's very hard, and funding is short, so people compete. And they weren't going to hear it from a little lady that, a foreign lady who had babies and who did this and
and work on glucose metabolism.
What the heck
and
was she doing? And matrices,
and the chick cells,
and chick cells, and floating this and that. And in fact, I gave a talk last week. Bruce Ames.
No.
You don't know Bruce Ames? Well, he was the most famous scientist in Berkeley in the 70s and 80s. And, he did the Ames test for carcinogenesis and whatever. And he was a very nice guy. He was a very close friend of Luigi Gorini, my professor. And many years later, when he had talked to a colleague, Richard Pito, actually, who is Sir Richard Pito now in England, had said to Richard, what is Mina doing? Why is she's so well educated? Why is she doing this messy stuff for? And last week I gave a talk at Children's Hospital in Oakland. He's 85, he was in front. Oh. And he's doing beautiful work still. And I said, I'm dedicating this to Bruce, and maybe he can see what I was doing. And he came and said, oh, Mina, this was wonderful guy.
So, Mina, in the early 90s, you started work on the human breast and the effect of extracellular matrix components on cell differentiation. Was this the time when Ori Peterson worked with you? Was he postdoc in your lab? I'm not sure.
Not at all. So, the first paper that really nailed the assay of 3-dimensionality with the role of matrigel. And it doesn't, they called it matrigel, and people just don't understand that this is just a surrogate for basement membrane, but you have to be very careful about how you use it. Because a basement membrane is not a basement membrane, it's not, it has to have the right properties, etc. It was a paper we published in Development in 1989. It's a very important paper. It was done in mouse, and it was done by Mary Helen Barcelos-Hoff. So she was my postdoc, and she did this very lovely paper. And in the early 90s, we did a series of experiments with a lot of people who came from different places. I had all these wonderful postdocs. I had Rabia, I had went out to Charles Stroili, went to
Alex Stoker? No, not Alex Stoker. Who's in the UK?
No. It's no. Andy Stoker.
Andy
worked on chickens.
Absolutely, I remember.
Andy worked on RSV. He was fantastic. He's now whatever. But the people who followed on this, Charles and I published a couple of very important papers. And then I had a fantastic graduate student in the 80s. In 1985, I have 2 very important papers. On using this glucose metabolism, and also doing a whole lot of other things, using this thing of Melvin Calvin. Yes. And that the person who did her PhD thesis with me was Eva Lee. And she is a very well-known professor and the chair of biochemistry department at Irvine. And her husband was Uno Ali, who discovered the RB gene and sequenced the human RB, and the two of them were a team. But she did her PhD in my lab, and we showed that it is the endogenous basement membrane that actually is doing something, okay? And we also showed that that when you give the basement membrane, the milk production is endogenous.
Yes,
so it's not just a stability, okay? So we did the stability studies, we did label studies, we did whatever. So extracellular matrix was signaling absolutely to new stuff, okay? And this is before we started doing RNA or whatever. But those papers are absolutely lovely. One in PNAS and one in Journal of Cell Biology, 1984, 1985. Then Mary Helen came and did that paper, and then she was a radiation biologist, and then she left, and then I had a letter from somebody called Ole Peterson.
It's a letter, of course,
I had no idea who he was. And he had written to Ruth Sager at Harvard, who was doing a lot of very nice mammary cancer stuff in those days, and to me. And he had said, I want to come and collaborate with you. He was a young assistant professor in Denmark. And Ruth apparently, luckily for me, didn't answer. And so I wrote and I said, absolutely, I want you to come because I have something very important in mind. And so he came to my lab, and he's thoughtful, he's an MD PhD, he takes his time, he's like Rick Schwartz, my model of how people should really deeply do science. So he came to my lab, he was very young, he was, and he had a graduate student who also came with him.
Oh, he came with his graduate student, that's...
Well, yeah.
Like a sabbatical or...?
No. Initially, he came and we just sat down and talked, and then later they did a 2-month sabbatical or whatever in my lab. They weren't doing any molecular biology, they weren't doing any cell biology. Well, they were doing cell biology, but they were doing everything. They were basically putting cells in tissue culture plastic and doing immunofluorescence, And so when he came, I said, Ole, what we really need to do is we need to show what we showed for the mouse, whether it is also true for humans. And the other thing we need to do, we need not just do normal. We now have to ask the following question. If the normal remembers, when you put it in 3D, as in the paper with Mary Helen and a paper before Mary Helen, when we use in PNAS in 1987, where we put laminin, type 4 collagen, dystroglycan, blah, 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 on, and then we put the cells on top, and we could see that gene expression went up with dot plots in those days. So that was before Mary Helen. But Mary Helen did the first nice experiment inside the gel with this, and I said, we need to do this, and we need to do the tumors, because nobody has an assay for epithelial tumors the way they had for fibroblasts when they dumped the virus on it. Okay, you dump the virus and you get a little thing, a little focus, and you count it. And we don't have this. We inject the tumor into the, you know how it was in those days. And I said, if the normal remember to be normal, maybe tumor remember to be tumor, and we can have a gorgeous assay.
Yes, this is so important, given that 80% of cancers are carcinoma. One cannot study, back then I guess people were studying cancer in fibroblasts.
Exactly, they couldn't study it in a way that would actually look like cancer.
Absolutely. Okay.
So we did this very lovely paper, 1992, in PNAS, and it's the very important paper because first of all, it shows that when you put the normal cells, the way we also showed in '89, into these cells, they stop growing. Okay, and they make this beautiful organization. And in the mouse, we had the milk.
Yes.
In the human, we don't have the non-pregnant humans that have reduction mammoplasty. Is not easy to make milk. You have to do other things, and mice and human have differences. Okay, But so I said to Ole, we need to find new markers, and the markers should be markers of polarity. So we should look at the basement membrane formation, and because we really should stop calling it matrigel. Matrigel is just a trade name, you know. Mm-hmm. It should be a mixture of a gel which has a certain characteristic and has laminin in it.
Sure.
Laminin, we have shown over and over again, is one of the most component of tissue specificity. So I said that let's look at the basement membrane, and when we did that, in the mouse matrigel, we could now see that there's a human basement membrane form.
Sure, then you can see.
And you can see the basal polarity magnificently. And the apical polarity, we did sialymucin, and we tried to do few other things for apical polarity is much harder to see. And we saw that it is, and I postulated then that it is the endogenous basement membrane that actually gives the tissue specificity. Because you put liver cells, as my husband had done, Monty Bissell for liver, you put it on matrigel, they make albumin. We put mammary cells in them, they make milk. Yes. So where does the specificity come from? The specificity is the information that the gel gives to these to form what they should form.
So Mina,
yes.
It took a long time before 3D cultures became used in many labs. Mm-hmm. In his book, The Tipping Point, Malcolm Gladwell says that connectors, mavens and salesmen are responsible for starting word-of-mouth epidemics. Why 3D cultures didn't tip like epidemics 20 or 30 years ago? Is it the connector, the maven, or the salesman that was missing?
You're being very literary. Okay, so I don't know why. I think the question of the fact that context is the most important thing, and that the phenotype is dominant over genotype, should not surprise anybody. And now that I say it, and I have said it for 35 years, a lot of people say, but it's so obvious. Why didn't we think about it? The reason is what I just was telling you, that the excitement of the time, and also I think something has happened in biology which is not healthy. Mm. And that is the fact that it has become a big money maker. And a lot of people find companies, and as such, they are then become wedded to their ideas. They don't keep their minds open. And they are huge amount of power and huge amount of money is involved in being right. So the first thing they do, they kill the messenger. And that's the idea of, and if I weren't as stubborn as I am and wasn't raised the way I was, I probably would have given up long time ago. And it was very difficult. It was many years without NIH money. Now losing my merit after all these years, when I get a merit, the head of the National Cancer Institute decides that he doesn't like the program, but he also takes away the merit that was already given. It absolutely boggles people's mind, and it boggles my mind. But it's very discouraging to see that people want to be right, and so therefore anything else that doesn't fit, they say it's against paradigm. And you would think that the best scientists would love to have a paradigm broken. Sure. That is the whole essence of science. But people have to be right to make money. They have to be right to do whatever. So you try to say, and the other thing is, of course, this fever to win the Nobel Prize has always been there. Has always people, you go back and you see how people messed up each other. Science is no different, unfortunately, than a lot of other professions. Is that there are still many good scientists who spend their lives because they're curious, they're passionate, they want to cure a disease, and it's not just about me. Now, I say I did this and I did that, but the reason is that I had to insist in order to be able to get people, train people. But the biggest and the nicest thing I have done, that of which I'm very proud, is the fact that I have trained a lot of
Yeah.
fantastic people, all of whom are doing so well, and they're spreading the word, they're showing it, they're doing it in a nice way,
Sure.
and it's a great, great honor to have had this. And I learn from them, they learn from me, and I'm very happy. And I think then we started bringing some other people. It took me 6 years to get Joan Brugge to come and say, okay, I now want to learn this. And once she learned it, she saw what a big difference it makes. And but other people were not willing to do it because it's harder, it's more expensive. But once you learn it, it is robust.
Absolutely.
And it's wonderful.
Absolutely.
So that's my explanation, and I think that we need to be mindful of people who have companies. Really. And I think that these money corrupts, and absolute money corrupts absolutely. You can buy things, you can push things, you can do whatever. And power does the same thing. So I think that as scientists, we really have to watch out that we separate ourselves from these things.
Absolutely,
So that
yeah.
we can actually appreciate and open our eyes and not always look under the light. And the biggest advice I tell people, I don't know if you watch my TED Talk, but
I did, yeah.
but only 17 minutes, and I had to talk to 600 people in the audience, none of whom were scientists. And one of the things I always say is, just don't get arrogant, because arrogance not only kills curiosity, which is what I said in the TED Talk, but it also makes you do bad science, because you think you have to be right. And you are not learning anything. You think you have all the answers. And there is so much we don't know. And there is so much to discover. And it's such beauty in discovery. And there is so much we should be doing and we still don't know. And people are unwilling to accept it. They run around and they think, they said when they sequence the human genome, we would know everything. And as I said, I was the director when the genome was being sequenced, and the first thing it started, one of the very first thing under me and in other national labs. And I kept saying, it's great, but don't exaggerate.
Yes.
So you have to exaggerate, you have to sell, you have to do, and all of those should be foreign to science and scientists.
So Mina, you published over 380 papers. We will unfortunately not be able to discuss all the topics and major breakthroughs from your group. I'd like that we talk a bit about your recent work on how the perivascular niche regulates breast tumor dormancy. Can you tell us in a few words about implications of this work?
Well, I would love to tell you this, but I have 5 papers last year, every one of which were a paradigm shift. And the vasculogenic stuff that we did was really 90% from Cyrus Hajar's work. And I would like to have him a few years from now to be interviewed. And he would tell you that he would have probably not done that work in any other lab except mine, because he keeps saying it all the time. But he, and I think that people should read that paper because it's very complex. It's another paradigm shifting paper. It was on the cover of Nature Cell Biology. I'm very proud of it. But he can speak for this recently or now or whenever. It's important because it's the kind of stuff that shows the beauty of microenvironment. Because in the same microenvironment that we can have dormancy, we could also have tumors starting to grow. And we know the factors that are involved, and those factors are going to play a very big role in either keeping something dormant or making sure that when they are no longer dormant, we can kill them. It has all sorts of implication
sure.
for how to go about metastasis and dormancy.
About the minimal residual disease,
Absolutely.
which is really one of the key challenges
Absolutely,
in cancer
absolutely.
biology.
And I am so proud of all the students and postdocs last year who did all this wonderful work. But what I like to do is to just talk to you a little bit about our glucose work, because we spend so much time on early on during this conversation about the stuff I did in the 70s. So I had the fortune of having a Japanese guy come become my postdoc. His name is Yasuhito Onodura. And he's now back in Japan, an assistant professor. I'm actually having a hard time because, you know, assistant professor in Japan very often are used as a surrogate for what the professor wants to do, and they don't, you know, let
they
them.
don't give them independence.
Yes. And, but anyway, this paper came out in December of 2013 on the web and was published in 2014, 2nd of January. It's in Journal of Clinical Investigation. And finishing what I started in the 70s, it took me a very long time to have brought people to try and think that if we use an inhibitor of a signaling pathway in 3 dimensions in tumor cells, as you know, we can revert them.
Sure.
And that was so hard for people to understand, and I don't again know why. And we show over and over and over again that the architecture and the structure of the tissue is extremely important. And we show that if the structure is not that, the information that has to make your nose or your finger or your breast or your tummy or whatever is going to get lost, all right? So the stuff that I taught about and did in the 70s with regard to Warburg's has gotten very famous again. When people couldn't find simple answers on a single oncogene, now they're saying everything is metabolism again, and everything. I say everything is important. They all have to integrate, okay? So it took me 30 years to get someone in my lab to get interested in doing what I did in the 70s in now.
Sure.
And Yasuhito took the challenge, okay? So since we can revert the malignant phenotype, we use beta-1 inhibitory antibody, it brings down EGFR. We inhibit EGFR, it brings down beta-1. We do MAP kinase, it brings down both of them. We do this one. So we show that in order to form a three-dimensional tissue, in order for it to know what it's supposed to do, all the signaling pathways have to integrate. Everything has to integrate. If a cell knew how to grow, another one wanted to apoptose, another one wanted to become cancerous, another one wanted to differentiate, you will be a mess. You would never have an organ that actually works. So I said to people, and I said to Yasuhito, glucose metabolism has to fit into this signaling model. So we have one of the best models. We not only have developed, taken again from Ole Peterson, the first model of progression of human breast cancer, where we can make malignancy out of human, and we have done all these wonderful things over the years that we have done. But we now have a way of actually having the first model of a human organ, that part of which we know how it's coming together. And when my next paper comes out, is going to be really important. And I will tell you one word about it at the end. But this one we showed that everybody who is now brought back Warburg says, okay, you add an oncogene. The oncogene makes the cell malignant. The cells grow. They need energy. Glucose uptake goes up, and then that uptake of glucose is very bad for you and the tumor. We said, well, going back to what I told you earlier, could the glucose uptake itself, increasing glucose uptake itself, be an oncogenic event? And that if you increase the glucose uptake, then you should activate oncogene, the same way that the oncogenes activate glucose uptake. And that the thing is dynamic and reciprocal, right? It's not going one way. For crying out loud, why can't people learn this, you know? So one thing that he did was he started doing this. He found out that in this progression model, the malignant cells has 400 times the level of GLUT3, which is the glucose transporter. So if we now put the cells in 3 dimensions, and we ask the question, what would happen if we down-modulate glucose uptake? Okay, now remember, in the 70s, I had not answered the question of malignancy straight. I just had said patterns become similar. But now you're talking about actual malignancy as judged by the structure you form, because the tumor cells make this ugly thing and the normal cell make a real normal thing, right? Okay, so so he took glucose away from these malignant cells that they revert to a normal phenotype, and they revert magnificently, and he discovered what are the pathways that connects the increase in aerobic glycolysis or the inhibition of aerobic glycolysis to other known oncogenic pathways. So if you will, the increase in glucose uptake itself is like an oncogene. And we have discovered 2 new pathways that people even didn't know exist, and they connect them to each other, and the reviewers killed us. They kept saying, HIF should get activated, this should do whatever. Why? When we do it in 3D, that's not the important pathway. The important pathway are the pathways that integrate in 3 dimension. And there is nothing about you that is flat, that is in 2 dimension in your body. And it really is a wonderful paper. And again, I was very lucky to have Yasuhito Onodora, and it was a pleasure, and I will always work very hard to see him to have his own lab, to his own group, and make wonderful new discoveries. So I find this very exciting. And at the very end of our conversation, I will tell you an unpublished data, a little bit of it, and you will see why it's so exciting.
So, Mina, you mentored many postdocs and students. How important was this to you?
Very important. I mean, that's one of the reasons I am quite patient with the students and postdocs, to make them understand and think deeply about what they want to do. Every one of my papers that comes out, and it's product of 5 years of work, 6 years of work. And luckily, for a while, I had enough money to be able to afford that. Unfortunately, it's not as easy now, because some of the money has been taken away, etc., etc. But nevertheless, the mentoring is the biggest joy of my life. And if you look at the former students and postdocs I have had, you start way back. You look at Joanne Emmerman, she became the dean at Vancouver. You look at Michael Zuika, who was my graduate student. He is doing the most beautiful work if you look up in leukemias and stuff. He was my graduate student who did that wounding paper. If you look at Charles Stroily, Andy Stoker, you name them, and then Celeste Nelson, who is now at Princeton and did that beautiful piece of work, and Nancy Boudreau, who is a professor at UCSF, Valerie Weaver, who is a professor at UCSF, Derek Radisky, who is a professor at Mayo Clinic. I mean, it just goes on. And Cyrus Qajar, who is now at Hutchinson Cancer Center. And a bioengineer came to me. It just, her name is Sumin Lee, and she's now a professor at Michigan. And it just goes on, and these people are all well trained. They have learned to have the integrity you need to have. I mean, I must say they must have had it, because otherwise you can't instill that easily. But at least I have instilled in them the whole way of life that would make a life worth living. Yes. I mentor them not only in how they should but I also mentor how they should do experiments, but also how they should think, how they should perform, how they should present, how they should then train their own students. And it has been a great joy.
So can you share with us your thoughts on why there are still fewer women than men as faculty in many places, not in the US, in Europe?
Is context, and is the fact that we are not training our sons and our daughters correctly. Because I told you, in Iran, at this crazy time, I hear 52% of faculty are women in Tehran University. Now, that doesn't happen anywhere else, but is the culture, is the context. We have to go back to the most important element of humanity. When women start getting pushed around and they get victimized, civilization is taking steps backward. If they, God forbid, if we have to go through this with Afghanistan again, all these stuff has been such a mess. So I sort of think that everyone, not only women, but men and women, all of them have to work for a time when this men-women business is gone, and when we are able to actually take the name away, the face away, and look at the work. And a few years ago in Sweden, there was this incredible to-do in Nature article where people showed that if you cover the titles for the same kind of a research that gave a guy 100, for example, gave a woman a 20.
Yes.
5 times lower just because the name is a woman. Men are prejudiced deep down because they don't understand, they don't accept, the women act like men. But God, it's wonderful to have men and women together. It's wonderful, we're different. It's not as if we don't do as good as science, we just think differently in addition, We dare to ask questions that very often the guys don't, because they are raised to, be in a group and show themselves, and if they are too different, they get pushed out the way a whole lot of other people, men also, have been pushed out. But women don't have that much to lose, and when they come, and if you look in cell biology, you see some of the best cell biology work now. It's done by wonderful women, as well as men, of course, but the time comes when the men take this problem as seriously as some women. And the women take it seriously, you have to make them empowered, you have to give them trusting to themselves, they have to learn to have a sense of themselves, to not have inferiority complex, to not have fear of success, and it takes time, but we all need to work toward it.
Thank you. So, Mina, what would you advise a junior faculty who is going to start her or his lab to work on breast development and cancer?
This is a very interesting question. For years I go, and every time I give a talk, I also give a talk to the young people in a separate room where we ask the professors to leave. And very often they ask that question. And I always say, look, I don't know where you will have your job. I don't know who you are. I don't know the characters of your postdocs. I don't know all of those. But of course, it's quite important to have integrity. It's very important. Those basic things that you have to have and you have to do. And persistence. You have to have persistence. But there is one thing I can tell them that they should do. And it's universal. And I say to them, put your hand up if you're going to take this advice. And, Americans especially, they don't want to take advice before they know it, and I don't blame them. But I say, look, if you don't want to take it, you can leave the room. And I have never had anybody leave the room yet. Once, in fact, I had all the professors come after I gave my first talk. It was supposed to be in a room like this, and then so many people came, we had to go, and I made the professor put their hand up. And some of them needed it. So I say to them, exercise. They look at me as if I am nuts. But you exercise early on. You will have the energy and the clarity of head to be logical, to be able to not get exhausted. And everybody says, I don't have time. And when I was director of 600, 700 people, I had 2 kids, I had 4 grandkids, and I would exercise only because finally I got to a point of my life where my back was hurting and killing me, and my neck was hurting, and my husband was saying exercise. And people don't listen to their wife and husband. But maybe they would listen to their mentor. And once I started going to physical therapy, learning how to exercise correctly, stretching, doing whatever, all things changed. So exercise. And from my glucose paper, that if you take too much glucose, it's bad for you. So if you take your second cheesecake or your second creme brulee or whatever, think about it. Diet is crucial, exercise is crucial, and you can have a long life and do something good with it. As long as you don't get arrogant, you exercise and you eat well, then your brain will work and you'll be enthusiastic and you'll do good work.
And you can train your people.
Yes!
So, Mina, what are the major questions that you think still need to be answered in our field?
Well, listen, in the breast cancer field and in the mammary gland biology, there are hundreds of papers coming up about the stem cells, and nobody agrees with anybody else. The technology is not there yet. The way we think about things is not there. People are fighting about, I'm right or this person is right, whatever. We know very little about the origin of the tissues. We know very little about tissue specificity. We know very little about how an organ in your body talks to another organ in your body. We know so little about how brain and neurons go to the mammary gland. We know nothing about exosomes. We just discovered a few years ago exosomes, these little things that bud out. There is so much to discover. There is so much to know, and we are still going and sequencing tumors as if that is going to give us the answer. Because we know how to sequence, and now that we learn how to sequence, what else do we sequence next? Tumors. And then we find every one of them are different from everything else.
There was an editorial in Science Signaling by Mike Yaffe about sequencing.
Yes, I saw that.
And it says it's like the metaphor of the drunk and street lights.
Exactly. That's what I have said for many, many years. You look under the light. That's exactly what it is. And when I was director, I kept saying, don't exaggerate. What are you talking about? We know nothing about the language and alphabet of form. We don't know anything about, and recently a cell editor asked me to do one of these one-page snapshots of branching. So I said, you want the branching of mammary gland or you want branching? He said, of course, branching. I said, you know how many dozens of different ways tissues branch? Just because they have went, it doesn't mean they do it the same way, or just because they have whatever. I said, what are you talking about? People will learn nothing by simplifying to that level. The simplicity is the beauty of the complexity. In other words, we ought to understand what it is to understand emergent phenomena. We have to see how the context changes things so incredibly. So I now have done one, and what I did was that I went and got somebody in USC who is evolutionary biologist, and he works on feather formation. And so he's doing the feather, we're doing the mammary gland, and it's just only two tissues, and in between there are dozens and dozens of different ways to branch, and they have things in common, but they also have things that are so different. So there is a lot to learn and find, and this whole idea of how you actually use the microenvironment to treat with drugs, as well as the tumor, what it is you have to do this. And if people go back and read nothing else but Gil Smith's work, you will see that you can take adult testes and put it in the mammary gland, and it makes milk, for crying out loud. I used to say there is no terminal differentiation. What is it you're talking about? And people would think I was such an iconoclast. And one morning you get up and you have Dolly, you take a frozen mammary gland of the sheep, and that has all the information. So why don't you have 3 heads? Why don't you, all these things are so important. There is a lot of imagination, there is a lot of patience, there is a lot of persistence we need.
Thank you, Mina. So, what would you like to, besides exercise, what would you like to tell the next generation of scientists who will watch this video?
Well, I have said already quite a bit of my advice. I have said don't get arrogant, okay? I have said be persistent. I have said exercise. I have said worry about diet, etc. But I would say that they need to have also the courage of their convictions. And I think they have to trust themselves. They have to just not be awed by these very famous whoever it is out there who gives a lecture and leaves the second they give their lecture, and they don't want to even talk, they don't even want to share. They act like that so that you don't dare to say, hey, emperor doesn't have any clothes. You have to have the courage of questioning authority. You just gotta stop listening to every word of your textbooks. Textbooks are dead, okay? You have to actually go in there and look at your data and try to interpret it in the way you see it. And then you can go look at the literature, rather than when you see something different, you say, it's against paradigm. And you won't believe how many times in the last 2-3 years with the paper I have published, I have had to go to the rescue of a lot of people where their professors say it's against paradigm. Crying out loud, you know.
Wow.
So there is a lot to be done. And also, remember, you're in science because you love it. And if you're going to be good, you have to love it. But just because you have a PhD doesn't mean you have to continue to do research. You have to say, my PhD gives me an amazing capability, an amazing way of starting a project, writing it down, finishing it. It's just like going through 9 months of pregnancy, except extended, and then producing the baby. But once the baby comes out, there are all sorts of other things that needs to be done. So if you read that one page I was talking about, you know, the nature cell biology things, there are one page, John Brugge has one, other people have one. And I said, if they weren't all these wonderful people who helped me along the way, and because, you know, there were a lot of people who were mean, nasty, and not very good, and
context,
I would,
but there are,
and then if it weren't for them, I would have a Persian restaurant, and it would be a good one. Okay?
No
That
doubt.
is so important, because you have options. You can do other things. My daughter was making movies in Hollywood, and then she had a baby and another one, and she decided to stay home. And then at the age of 45, she decided, I'm going crazy. This didn't work, that didn't work, that didn't work. She went to graduate school, and now she's getting her PhD, and I'm going to see her in June. She wrote a thesis on children and media, because she has children and she was in Hollywood making movies. She was, executive, very successful executive producer. So she's using those data to do something else. So always have a second option. It is not the end of the world when you don't get your grant. I couldn't get my grant because people thought I was crazy. But I always found some people who believed in me and who were willing to help, including even just 2 years ago when I lost all that funding. There were people who came and said, we want to give you money. You're a good thinker. We want to help you. But if they wouldn't do that,
Yes.
I would be doing something else.
Sure.
And that's not the end of the world.
Sure.
Just live a life that allows you to do good. I love the motto of American Philosophical Society, and I am very honored to be a member, having been elected and a fellow. But their motto is, we're here to do good. I love
nice
that.
one.
Yeah.
Mina, is there any question that I should have asked you, but I did not?
Yeah, you know, this question of how people are having 5 minutes of fame these days, that people jump to publish, then people jump to criticize, people, you know, this whole reproducibility initiative that people are now making such a big fuss. It is very important to reproduce. It is very important to be able to reproduce. But as if people read that little article I published with Nelly Polyak and with each of our postdocs, Kurt Heinz, who is the first author, and Nelly's postdoc, very, very capable gal, it took us 1.5 years to solve a problem that could have been simple, but there are certain things that just doesn't even occur to you. And people go, we are following a method that was put together with Martha Stanford 12 years ago, and they are following another one that somebody else had developed. And we compared everything, and then we stood side by side, and then we solved it. And we now will help all this confusion in breast cancer, because a lot of it in breast cancer and mammary gland, because a lot of these things are done with facts. And people don't realize how many different steps before facts and after facts you have to do this. And in fact, I just met with the chief editor of Nature Cell Biology, who came to my office, and she was saying, you really should publish this in very much detail, because the confusion in breast, more than anywhere else in a stem cell condition, requires that kind of, So she said, you should go to the Nature Protocol and publish it, so maybe we will do it. But you really gotta think before you accuse someone of saying, we can't reproduce these data. On the other hand, the minute you work with this person and you find that together you cannot reproduce it, they have to withdraw the paper. And if you don't have the courage of your conviction to then expose it if they don't do it, then there is something wrong with you. Because there are a small number of people who, because of some psychological problem or whatever, or because they so badly want to be right, or because of whatever, because of pressure from their PIs, they should actually, do something they shouldn't do. The last thing I think to give away is, if you are the head of a lab and your postdoc comes from Japan or China or India or American young person or whatever, and you publish a paper, then later somebody says, this is not a good paper, it should be withdrawn, don't dump all the responsibility on the first author. The head of the lab, more than anybody else, is responsible for the content of a paper. You should own the problem. And that will stop people from then perpetuating this. Because there are a lot of professors who come in and they're so in love with their ideas that these young people want to please the professor, and they see what they see the professor wants, and sooner or later they, publish it. And the professors is very happy, they are happy to get the glory, but when some trouble arises, even when they withdraw the paper, they then don't take responsibility. And we should talk about that. And for the young people, you set the standard very high for yourself, and you should set the standards very high for your wonderful postdocs and graduate students. I mean, my wonderful postdoc and graduate student that I have had the pleasure and the honor to work with.
Mina, you mentioned you wanted to talk about some more data that are coming. Would you like to say a few words?
You remembered that. I was going to forget it. Okay. So the question, we have spent 35 years step by step saying, how does the extracellular matrix actually talks to chromatin? How does, and we have found, for example, actin in the nucleus, which is responsible for growth regulation. And we show that it's that pathway that goes off in malignant cells. We haven't published this. So malignant cells don't respond to laminin because whatever signal goes to the nucleus to get the actin out is not there. So we are going to be publishing this. But I have this one was a wonderful Brazilian postdoc who just got a fantastic job in Brazil, and I'm so proud of him, Alex. And then my Japanese postdoc, who is just a fantastic gal. Her name is Saori Furuta. And we're going to go out and publish this paper. She has been talking about it, so I will talk in the part of this stuff that she's already talked. We haven't yet published it, but we have sent it out for publication. How is it that the laminin out there has so much power to do all the things I'm talking about? People don't even, there are people who mix up laminin and lamin. So what can I say? I'm writing a review for Science with a few other people, with one of my new postdocs, which says laminin to lamin revisited. I wrote something in 1995 saying laminin to lamin.
Yes.
But what is it exactly that happens? Well, guess what we discovered? Laminin activates P53. And P53 activates laminin. In the dynamic reciprocity and in the formation of all of things, you have 7,700 papers in P53. Has anybody ever discovered this? You see how much there is to discover? It is so exciting. So we are closing the loop of the three-dimensionality about how these things have to come together and how we can revert the malignant phenotype and how we are doing it. So we are very excited about this. And as you can see, I haven't lost any of my passion about science.
So
okay,
Mina,
Momo,
thank you
that's
very
enough.
much. That was, thank you.
You're welcome.
That
My
was great.
pleasure.